from. Where is my selected? I already took for. No. So, there it is. So, this is the inoculation we didn't do at all during our practical work, but it is the simplest inoculation. That's why people, usually persons who got this inoculation, have no chance to make a mistake. This is so-called stub inoculation. So people, I already showed you the medium for stub, like ferric sulfide agar, long pole and so on. People, this is for real inoculation. This is for you, pole of agar. For you will be enough. Okay, let it be. So people, what needs to do? First I'll show, then explain. What are the potential difficulties? First of all, sometimes plugs might be greatly attached. If ladies have very small finger, little finger, for them it's difficult to operate. Yeah, something like that. So that's why it will be not a mistake if before taking the plug you little bit relax. So of course don't remove, little bit, left, right, some way. Move it from primary position, so to relax. So you may do it even without my observation. I'm turning back, you relax. Something like this. So, okay, next moment, people. Principal position concerns several tests. From which test tube, if two test tubes are in operation, should be below and into which should be above. So here, best of all, as you asked me what to do first, from which this one, into which this one. So that's why I adjust with, so I'll loop, I have no loop in my hand because it's difficult to do. So this way, something like from which, into which. So, and then after that, spirit lamp already on, sterilize the loop and open it this way. Of course, principal moment, little fingers.
Yes, very little fingers.
No, maybe.
No,
Okay.
could you do it again?
Okay.
Fingers.
So sterilize, open, sterilize. So, and then manipulate it. So people, again, so don't forget to cool the loop. Take very small portion of culture, and inoculation I will not do, but it is very easy. The easiest, simply penetrate up to the bottom. One movement. Here, up to the bottom, and then remove. Sterilize, close, sterilize, put inside. That's all. Only one explanation, moreover associated, as I already began to speak about, ferric sulfide for cultivation of anaerobes. Which anaerobes we already said about. You will make comment that it's better to cover it. First of all, you may say that pole of agar should be much more longer. It should be covered with Vaseline oil for better isolation of anaerobic bacteria, and so that's all. Understood? Let it be so. So let me remove maybe this pole back. This also. So what comes next? Let me look. So what will be more? So planting of material, liquid medium little bit later. Oh, planting of material on petri dish with MPA. It's initial inoculation. So usually will be given liquid medium for as source of microbes. Oh, maybe from here. So and I will give you free petri dish. From here. So people, again, like classical versions will be shown for you, because people, I think every microbiologist makes personal kinds of manipulations, but we demonstrate you something universal. Making horizontal line again. So this horizontal line not greatly mandatory, but better to do it. So putting upon the table. So sterilizing the loop. Sterilizing the outlet of test tube, opening, taking the drop, sterilizing, closing, then people, the rest. As I said, I recommend, if you got inoculation, especially into petri dish with medium, check your, look critically at your loop. This is okay, by the way. I have nothing against this one. Do I see here something rigid version? No. Here in this class everything looks normal. It's okay also, my opinion. It should, it doesn't matter. I don't want to confuse you a lot. So here it is. Loop of culture is here, usually several microliters, up to 5. So people opening this way. Of course, I will do no violation. It's -1 will be if you open this one. Don't laugh. Such case is found. So that, and then parallel streaks. Again, various versions. 1 streak, second.
See, from outside to inside.
No, as you like. By the way, if you are left hand working, maybe for you more comfortable this way. It's not greatly like indicating. One streak, put up, second streak, third streak, up to half. So principal moment, the material should be the same. Don't take material second time. Then turn over and again, from pole to middle part. Totally 19 streaks will be found. Distance between streaks, try to make them parallel. Distance between streaks for about 1 centimeter. For about 1 centimeter. Lesser possible. So 10 millimeters. So people, inoculation is done. What can I ask here? First of all, try to remember something like this. Not so much information. So people, all these manipulations, previous one, that one, next one, will be used to isolate pure culture. That's why people, you have to revise some definitions. What is pure culture? This moment. If you want, you can make education. Next moment, people, to isolate pure culture. What will grow after incubation? What will appear? Colony. So here, let's make some balance. So I may ask you, of course, to revise all characteristics of colonies, starting from sizes, finishing with consistency, inner structure, and every one of them has several grades or something like this. People, at least revise S and R forms of colonies. So as smooth colonies, they're mostly wet and something like this, and rough colonies are regarded as dry, warty, and so on. So that S and R forms of colonies that will grow. And what is and of course I will ask, what is colony? This is the isolated group of bacteria of single species grown on solid nutrient medium and which are offsprings of single cell. So aim of all this test to isolate pure culture. Pure culture is almost pure culture is a group of culture of bacteria of the same species grown on any nutrient medium. Okay, let it be so. Something like this. So what is the next inoculation? So it was question 4. Now question 5 we discussed. 6, planting of material on slant agar with MPA. This is easy to do. So people, it might be inoculation. By the way, one trick, just a second. It will be anyway from Petri dish. Here we continue looking like we continue the isolation of pure culture. I need only 3 here it is. So, people, you will be provided with this Petri dish with colonies. Again, you have to encircle the colony. As for slide making, taking of culture will be totally the same. Sterilizing, cooling, taking the half of colony or something like this. Then close. Take the slant, opening the plug, sterilize, culture is here, and you remember, zigzag, left, right, up to the end, sterilize, close. That's all. The questions will be the same. Okay? Let it be so. Possible to remove this one. So, next goes, last to my mind remains question 7. Inoculation of material into liquid medium from slant agar. So, here it is. Continuation of previous version. So I will give you free. Here it should be. Here it is. Browse, just use with browse. Clean. So and slant with growth. Again the same. Here it is. You see sometimes different material of plugs. So that's why I am doing like you. So, relaxing this way. From which bottom into which above. Sterilization of the loop. Opening. Sterilization, of course. Taking small portion of material and put inside. By the way, it's not a mistake, but sometimes, generally no need to do that. Material is here, visible. And student wants to drop it until it disappears totally. No need, simply put inside, left, right. Sterilize, close, and the rest of material burn away. Is it clear?
Doctor, from this to this.
To this. Okay. From this to this. So inoculation simply immersing inside. Okay? Put deep and remove. Okay. Let it be so. People, but it is only half of the question. Now first I show you here demonstration. then you'll sit and write something down. In most of situations, inoculation from slant into liquid medium is used for detection of biochemical activities of microorganisms. So that's why, please. Okay. Of course, they should be revised. I will tell you about what to do. You will be in this question. First half will be done under my control, but I may provide you even before start of your performance. I may provide you with these results of inoculation. So here's a YAS so-called his rows or fermentation. I hope. Oh, maybe here. I hope fermentation visible. Maybe white background possible to apply. Fermentation is visible. Oh, just a little. Oh. So people, by the way, here it is, you very seriously like look at this one. Your task will be to describe in general. That's why revise his media. His media, it means liquid media for fermentation. And principle of its action. By the way, we already jumped ahead, like McConkie agar, principle of action will be totally the same. It means, so we operate with short his row, and here there are 5 sugars: glucose, maltose, mannitol, sucrose, lactose. By the way, they're all indicated, but in Russian. So, and initial color is yellow. The medium contains broth, indicator fuchsin, decolorized, and sugar. Microbe ferment sugar, acid products will be finally accumulated, and change of color indicator due to the decrease of pH. This one. So people, but you see here 5 sugars, but here's how many colors are visible? 3. Yellow, black and red. So people, pay attention on black. It like discriminates between last two wells and first 5. First 5 are for sugars, easy for them. You see, by the way, microbes are already indicated. Staphylococcus fermented all sugars. E. coli, sucrose not. All the rest, including lactose, also fermented. Remains the rest of 2. They are not for fermentation of sugars, they are for proteolytic activity. 2 basic end products, indole and H2S. H2S giving black color for Staphylococcus. Indicator lead acetate. For indole, indicator is oxalic acid. Also various versions of indicators possible. Okay, your task will be to explain that. The basic comment, why do we do that? To detect biochemical activities of microbes. And why do we do detection of biochemical activity? Because it helps to discriminate between various bacteria due to the different spectrum of their biochemical reactions. Okay, let it be so. So, was it a sit please back? I will now gradually come into serological test. I will also tell you about what to revise. Yes? Here is.
This is for number 5 also?
Yes.
Both we have to do.
Sorry. No, for number 5.
Streaks. When we did this.
No any stick. I will give you simply the pole of agar. You will remove it. Stop inoculation. You will make deep agar and remove that salt.
Okay, and the streaks.
Streaks it goes for question number 4.
No.
Of course. So now listen to. We came to serological tests. What to revise about serological reactions? First of all, again, it's previous semester book. So people, preface or maybe common chapter devoted to all serological reactions. 2-3 pages are there. First of all, that they are highly specific, highly sensitive. Why explain? Because epitope reacts with paratope. So then, I don't ask you to write down mass action equation, no need. So, okay. So there are 2 phases of reactions: specific that goes rapidly, and non-specific that demonstrates visible manifestations of the reaction. So, what else? 5 groups of basic groups of serological reactions according to their type, starting from the precipitation, agglutination, lysis, neutralization, and labeled antigens antibodies. This one. And basic reagents. About reagents I will speak a little bit later. So, people, which reactions to revise? Precipitation, agglutination, microbial and hemagglutination. No need to revise Coombs maybe or something like. So some details because we will not do them during practical skills. And labeled antigens antibodies solely ELISA test. So that's why I revise them. So I will, when speaking about them, precipitation asset, agglutination set, gamma-agglutination set, and ELISA test. Okay. So people, in the general description of serological reactions, you may see such positions. First of all, what does it mean, serologic or serological diagnosis of the disease? Serologic diagnosis of the disease.
Finding antibodies.
It is diagnosis of the disease by detection of
Antibodies.
So, as we many times discussed, possible to detect both hepatitis B or antigens or antibodies. Everywhere will be serologic diagnosis, but in most of cases antibodies. So that's why it looks like indirect diagnosis, not direct by isolation microbe, but indirect diagnosis. Okay, keep that in mind. So that's why forgot about the beginning. In every serological test, one reagent is known, second unknown. In this case, in serologic diagnosis, serum will be unknown antibodies, and we should use some called diagnosticum. If diagnosticum used antigenic, it might be microbial cells, erythrocyte antigenic diagnosticum. I will speak a little bit later. Okay? So people, here, again my favorite reaction. Where is it? Ring precipitation reaction, test number 10. So as I say, it's okay. By the way, everyone demonstrates me during class the persons. It means to spur the person and
It
person.
doesn't work for me.
So, okay, I understood. I will not look at you. I will look at someone else. Okay, let it be. So people, there were no exceptions. A lot of persons got this reaction, and everyone may be sometimes explaining poorly, but they may demonstrate me very, very nice, perfect, almost ring, narrow, without problem. So that's why will give you reagents, and you will make these reactions. Then we'll discuss about the result. As I said, the result was anyway, I must scare you. If you'll show me the ring, it will be, to this moment, no losses of points of your score. If you'll show me something unclear, not transparent, it will be 5, 6, 4 points you already lost. If you'll show me nothing, it will be 2 for this question, no subject of discussion. So, okay. Let it be. So people, this one. So, if to read carefully, what I may ask directly on the way. So, ring precipitation reaction to determine microbial antigens in cerebrospinal fluid. What can we guess here? Of course, serum will be known and the antigens will be unknown. If we speak about cerebrospinal fluid, what are the potential microbes? And what is the potential disease?
Meningitis.
What are the potential positive agents?
Meningitis.
Meningitis. So people, and I don't want you to go deeper into bacteriology, but second position. Who remembers what will be the microbe number 2? If not meningococcal meningitis, something else. Pneumococcus, by the way, pneumococcus. People, next it will be Haemophilus influenza, next it will be Streptococcus agalactiae, next will be even E. coli and anything you want. So, but meningococcus number 1. So that's why. Principal moment, precipitation test is made with soluble antigens. So both reagents should be colloidal solutions, soluble antigens. As a result, will be formation of insoluble immune complex. So you know that first you have to drop serum as the mostly dense. I will indicate very serum, very cerebral, and then apply cerebrospinal fluid. There will be no controls in order not to waste time. Free test tube will be given for, and you will show me what needs to explain. I will ask you, for example, how to prepare precipitating serum. As serum contains antibodies and it is diagnostic serum, every serum is made by immunization. If here it is diagnostic antibodies, it will be polyclonal antibodies, it will be laboratory animal immunization. Give me the example of laboratory animal. Rabbit, of course. So that's why immunization of rabbit means meningococcal, we say direct meningococcal antigens. Meningococcal antigens and making serum and making antibodies. This is one. Then I like to ask mechanism of precipitation. As we said, both reagents initially soluble, making insoluble agglomerate. something like. Here they are antigens. Usually antigens have not single, at least epitope. There are many epitopes. What do you think? Which class of immunoglobulins I schematically demonstrated? G, right. Principally two paratopes, at least two paratopes, two FAB fragments, two active sites. So people, I will not ask you. You are sitting far away from me. For me, myself will be more easy. One paratope goes to one molecule, second goes to another, one to another, making endless, very large agglomerate here. Also, how to draw? Oh, maybe one antibody goes here, one more molecule, something, and so on and so on and so on. Is it clear? Okay, let it be so. This is precipitation. Next, agglutination. And if you remember, the reaction is very rapid. It finishes in 1 minute, so you will see everything. So, next goes agglutination test. Agglutination test. So, it's number 9. Slide agglutination test for microbial identification. Who remembers cry of my soul? Don't draw ovals. So, here it is. So, people, unlike slide making, these serological tests are regarded as tests, no period. Okay, simply and incorrectly are done in non-sterile conditions. Modern approach to do everything in sterile conditions, in boxes and so on, but people, in reality not. So that's why. In this reaction, you have to light spirit lamp only when you take in microbes. It will be last step of this test. Before that, you have to prepare a slide. Taking the slide, simply remove the dust. No need to draw ovals. So, divide slide into two parts. So, and one part you may indicate number 1, number 2. Control experimental serum, saline, anything you want. You may indicate somewhere in the corner. And people put upon and drop, if you remember, 1 Kopeck drop of saline will be given for you and serum. Oh, by the way, unlike slide making, of course, we need dark background. Everything should be done upon the support. Honestly speaking, last step can be done upon support, but better to begin upon it. So then, only after that, you will light spirit lamp and take microbial culture. If you remember, it should be, as the result, milky suspension of both control and experimental part of your drop. So people, usually 1 loop of material in one side of your slide, not enough. As someone already asked me about, materials here you put inside, spread in the drop. Take in new portion, sterilize the loop. Put, spread. Take a new portion, sterilize the loop. If need, take 5 times, as you like. No, my recommendation, because sometimes greatly different, the density, like visible density of left and right side of your slide should be like equal. No plus minus. It shouldn't be that one is totally transparent, second is totally turbid. Okay? It means different amount of microbes were taken. Okay? Let it be so. This is performance. So then again I will ask about mechanism of this reaction. So agglutination, who remembers, is not simply clumping or binding of something, but clumping of particles. Role of particles here play microbes. So microbes, that's why revise basic microbial antigens at least by their names. O structure, in brief, H, flagellar, and K. Okay? Antigens protrude from microbes. Antibodies directly bind principle is the same manner. Something like and so on. Is it clear? Principle like in precipitation. By the way, forgot to say. That's why people in precipitation and agglutination, both next will be hemagglutination as well. Reagents should be not only specific but multivalent. For ELISA test, it's good if they will be multivalent, but not like it is critical point. Sometimes they might be monovalent and so on for certain reasons. So this one. What else? Let me read the contents. So slide agglutination for microbial identification. What is unknown here? Antibodies or antigens?
Antigens.
Antigens, of course, because microbe is unknown. So that's why people we use second reagent will be known agglutinating serum. How to prepare agglutinating serum? By immunization of rabbit rabbit with corresponding microbe. So that is. So people, closer to it will be hemagglutination. In number 8, indirect hemagglutination for serologic diagnosis of the disease. Okay. Oh, by the way, it should be primary recommendation. Before doing something, read carefully the title of your item. So that's why people, serologic diagnosis, what it means. Detection of undecided antibodies. So, patient serum will be first reagent with unknown antibodies. Without revising, students, almost everyone forgot what will be the second variant. Indirect hemagglutination. Unknown antibodies, what will be known? What is the name of reagent?
Erythrocyte diagnostic.
Erythrocyte antigenic diagnosticum. You may drop some sentence that erythrocyte antigenic diagnosticum can be obtained, can be manufactured if to take, possible say, neutral erythrocytes like cattle, sometimes even O group of humans, but better take cattle, with chemically absorbed necessary antigens. Antigens might be of various origin, depends what you want to do. What was done with serum, initially I will say a little bit later, and principle of reaction will be totally the same. Clumping of multivalent antibodies with 2 or more erythrocytes in erythrocyte antigenic diagnostic. Okay? Let it be so. So people, now I try to show you, even I don't ask you to stand up. This is the necessary for us people to take. I hope it will be easy. So, also, familiar reaction for you. How many wells are visible? 5. I hope everyone sees, right? So, what was done in serum in this row? With serum in this row? Dilutions. What happens with concentration in dilutions?
Reduces.
Decreases. So that's why. And if we have 5 wells, maybe 6, 7 wells, doesn't matter. If 5 wells, what is the last well about?
4.
Control. What is absent in control? No antibodies, no serum. So that's why we stop titration at pre-last well of test tube. So that's why people. And after checking of control, if control has no serum, will be hemagglutination or not? No, of course. So it should be like dot. This way, look. Do you see the dot? I think no need for you to stand up. Everything looks visible. So from last, you jump into the first one. How many wells are positive? 3. 3 wells are positive, making so-called, disliked by me, what? Umbrella. So that is, people, your task will be to determine titer. Well number 3 will be titer. That's why please revise the definition for titer. Titer might be for any kind of reaction, but here it is typical for hemagglutination. It's maximal dilution of serum or antibodies or minimal the amount that still produces positive reaction, positive hemagglutination test. Okay? Let it be so. So people, ELISA test now goes. So people, revise ELISA test, of course, yes. And let me read, again listen to, like question number 8, sounds the same. Evaluation of ELISA test for serologic diagnosis of the disease. What it means? What it means? What it means? Serologic diagnosis of the disease, detection of antibodies. So that's why people, we have to make scheme of detection for antibodies. So I think demonstration I will show you a little bit later when I'll show you all demonstrations together. So in order not to ask you to stand up many times. Anyway, I have to bring the picture with PCR next. Okay, so people, we are looking for antibodies. So if you remember, here it is solid phase, also known as heterogeneous immunoassay. As solid phase, we use polystyrene wells. If antibody is looking for, antigen will be absorbed. What should be added next? Antigen. Patient serum. If contains antibodies, it might be bound to AB. What will be the total?
Immune complex.
Immune complex will be formed. So, patient, I hope, is human. What should be added next?
Anti-human.
Anti-human immunoglobulins coupled with enzyme. How to prepare anti-human immunoglobulins? Enzyme peroxidase. So how to, by immunization of rabies with human immunoglobulin fraction. Okay, and these antibodies, anti-human, should be coupled with enzyme peroxidase. substrate for it hydrogen peroxide and chromogen indicator. So people, you may add orthophenyldiamine or tetramethylbenzidine, more popular now, TMB, or orthophenyldiamine or tetramethylbenzidine. So people, which changes do we observe after the end of the reaction? A reaction will be terminated by sulfuric acid. Optical density, which we see the change, the color equivalent to optical density. Where do we measure optical density? Colorimeter, photoelectrocalorimeter, or photometer, or multi-channel, multi-scan reader. Okay, so people, this is very popular question, and sometimes parallel to students may get this question. That's why, in order not to repeat the same for different persons, for second person, I'll ask how to reconstruct the principle to detect antigens, not antibodies. What should be placed upon the bottom of it?
Antibody.
Antibodies. Next,
Antibody.
antigen from patient's serum or any other biological fluid. Then,
Antibody.
again, antibodies resulting in which version of reaction? Sandwich immunoassay. Then anti-immunoglobulin antibodies and so on. Is it clear? Okay, people. Maybe how to do. I want to show you all the demonstrations together. Wait, I will bring PCR.
நல்ல காலம் நிறைய 10 ஸ்டார்ட் ஆகும் 10:40
தியரியே படிக்கணும்
ஆன்டிபாடி
இது தான் when things are going antibiotics or something like
so that Elisa he'll just give the plate
yeah we have to explain
in the plate he'll give Elisa we just explain this
he didn't tell you he thought of showing all the demonstration after